Yes, I'm here. Good morning or good afternoon.
Good morning. Okay, here's the afternoon. Yeah. Okay, you're on, so we can go. Oh.
Okay.
Okay. But I just check so the audience is with us, or? Okej, då verkar det som att alla är uppkopplade, inklusive USA här, i form av Marla Spivak som är professor på nästa universitet, som jag sa, och sysslar väldigt mycket med propolisfrågor och vaxfrågor till exempel. Hon kommer att berätta en hel del om rätt så spännande tester som hon har gjort bland annat. We give the word to you directly, Marla.
Would you like me to begin?
Yeah, you can begin now.
Okay, wonderful.
You put up the presentation by yourself, or shall we do it?
Yes, I can. Yes,
okay,
yes, no
okay.
problem. All right, well, it's morning here, but good afternoon there. I wish I could be with you. I know it's a lot warmer where you are than where it is here. Today here in Minnesota, where I'm from, it's in centigrade, it's minus 25.
Sure.
So it's quite cold.
We have summer here. It's just 2 degrees here.
I know, minus 2. I looked it up. But I'm showing you in my background, if you can see, this is the Mississippi River in the fall time, of course, not now, because now it's covered in snow and the whole river is frozen. In the background is Minneapolis, the city that I live close to. So it's a beautiful state, and I think it's kind of like Sweden. But...
Yeah, you know that when we left Sweden 100 years ago, it was Minnesota was the main object to go to.
Yes, there's many Swedish, many Scandinavian people that settled.
We have a lot of Karlsson and Larsson and so on.
All right, okay. So I'm going to share my screen and begin.
Yeah, there we are.
And you can see it now, right? Wonderful. I'm going to talk to you about something that's very near to my heart to these days. That we've been studying the benefits of propolis to honeybees for some time now at the University of Minnesota in the lab. And I'd like to point out our website down here, blab.umn.edu. If you browse around and go to information from my lab, you'll see a lot of the publications. They're all, you can find them all on our website if you're interested. So propolis collection, and I'll say that propolis can be pronounced propolis or propolis. Like sometimes we say potato or potato or tomato or tomato. I don't think there's a correct pronunciation. But propolis for bees is a form of social immunity. And this is a term that Dr. Sylvia Kremer from Austria coined in 2007 to describe behaviors, organizational strategies, and physiology that many social insect societies use to defend against parasites in order to maintain health of the colony. So honeybees and ants and termites and some of the social wasps have behaviors and other strategies to keep themselves healthy. So propolis is one of these strategies. It's a form of social immunity. And it's interesting because propolis is very sticky, and as beekeepers, you probably don't like it very much, maybe. And I'm hoping to convince you in this talk. By the end of the talk, I hope you're convinced that propolis is very, very good for bees, and we should start to like it and encourage our bees to collect more propolis. I know it's on all of my clothes and stained permanently, but now I've learned to like it. So propolis is actually resin, and I'll explain. So some plants produce these resins. These resins are plant defenses. Some plants produce them to protect the plant itself. They protect the plant against diseases and against UV light rays, and also against herbivory, being eaten by other insects, or other animals. And so the bees are able to locate these particular plants that produce resin, and they scrape the resin off of the plant. Usually it's on the leaf bud of the plant. So this bee in the center is actually from Brazil, and this plant is Baccharis, and she's collecting what they call green propolis. It's been studied a lot in Brazil. This is a bee from Minnesota, and this yellow resin on her legs, she's collected from, I'll tell you how we know this later, but from populus or cottonwood trees. And another kind, this red propolis, we know comes from balsam poplar or Populus balsamifera. So the bees collect these resins, they put them on their hind legs, and they carry them back to the nest, and where they cement them into the nest cavity. And once it's inside the colony, we call it propolis, because propolis was discovered long before we understood that it was actually plant resin. So early discoverers of propolis thought that it was produced by the bees, and that they understood it was medicinal, because we've been using propolis for human medicine harvested from honeybee colonies since biblical times. So when the bee comes back with resin in this middle photo, often it's so sticky, she can't get it off of her hind leg by herself, and another bee has to help pull the resin from her hind leg. And then they start cementing it into cracks and crevices within the nest. This photo on this side, where my pointer is inside a tree cavity where honeybees nest naturally in the wild. And inside, they coat the whole interior of the tree with this propolis lining. I'll show you another photo. As far as we know, the bees are not changing the chemical composition of the resin. They can add beeswax, and this is supposed to be an S here, not beeswax, beeswax, but they don't secrete anything into, that we know of, any enzymes into the resin to change its chemical composition. So this is a tree cavity that we found that was full of bees here, that was cut off. And here's the entrance to it. And here's when we opened it up and removed all the combs, this is the interior, and this whole brown lining is the propolis envelope that was termed by Tom Seeley. So this red line from a diagram from a book that he wrote in 1985 describes where bees deposit this propolis lining in the feral colonies that he studied in upstate New York. And you can see that it lines the entire nest cavity, and particularly around the entrance. So this photo on the right is that same tree that was in the previous photo, but this time we took a photo as if you were a bee looking out the front door. And you can see how much propolis they deposit, particularly around the entrance of the nest. All of this orange and yellow, sometimes green. It's a very, very thick lining. And here's the beeswax. So that's very thick in a tree cavity, but in our beekeeping equipment, bees do not put this propolis lining because we give them wood that's planed very smooth. And on a smooth surface, they tend not to put propolis. They prefer to put it where underneath the frame rests, or between the boxes, everywhere where it makes it difficult for beekeepers to manage the colony. That's why we use a hive tool, because the propolis is so sticky. Of course, some colonies produce much more propolis than others, or collect more. You'll find it in cracks in your beekeeping equipment. Here's a colony that really brought in a lot of propolis. It's kind of almost melting over the edges. I'm sure you've seen colonies that in the fall, before winter, almost seal up the entrance of the colony with a lot of propolis. So they have just a small entrance. And sometimes if we give them a thymol or a formic acid treatment, they'll completely coat the treatment with propolis. They'll also coat mice if they get inside the colony and die, and the bees are not able to remove the whole mouse, they might encapsulate or entomb the whole mouse in propolis. Some subspecies deposit a large amount of propolis, like Apis mellifera caucasica and scutellata are very known, those races of bees, for collecting a lot of these resins and depositing them in the nest as propolis. We don't know how much of these resins are incorporated into wax comb. We know in a feral colony, a wild colony in a tree, where the combs touch the tree, they put propolis here, you can see it, and they line the tops of the cells with propolis. But the brood combs in the center, we don't think they put propolis in those combs, but we don't know. So resin foraging is really demanding, energetically demanding for the bees. There's no direct reward from it. They're not eating resin or propolis. They're not getting it from the same trees or flowers or bushes where they would collect pollen and nectar. So they're flying to separate plants to collect this stuff. So why do they collect it? If it's just for cracks and crevices, they could use beeswax to seal up those cracks. So many years ago now, we started asking these questions. And down here are photos of the 4 graduate students who have gotten degrees trying to answer these questions. What is the benefit of a propolis envelope? Why do bees collect and make this propolis envelope? How or does it affect the honeybee microbiome? Another typo, excuse me. Do honeybee colonies with a propolis envelope inside the nest do they have less disease? Do colonies self-medicate? In other words, if they're sick, do they forage for more resin, as if they're foraging for more medicine? And what are the plant sources of resin in our area? So these students, I'll bring them up during the talk, and this was all funded either by National Science Foundation or the USDA here in the United States. I didn't say, and I should make it very clear, these resins that the bees collect are highly, highly antimicrobial. The chemistry of the resins is very complicated. It's full of flavonoids, phenols. In the tropics, it's mostly terpenoids. And they're variable, and there's very many of them. So there's not just one component in the resin that's antimicrobial. There's very many of them, and they're constantly changing from tree to tree. Even parts of the tree have different antimicrobial activities, different leaves do. So it's a good strategy for a plant to have highly complex antimicrobial properties and activities, because it would be effective against many diseases and not just one. Okay, so to start studying with the benefit of a propolis envelope to bees, this is Mike Simon Finstrom, my first graduate student to study propolis. And the way we started, which I don't recommend that you do, but I just wanted to explain, is we collected propolis from other colonies, and we extracted it. We made an extract in ethanol. So we dissolved the propolis in 70% ethanol and made a 15% solution. And then we painted it in the inside of these small nucleus boxes. So this one in the middle, we used that green propolis from Brazil. This one that's red, we used propolis we collected from Minnesota. And this one is a box that we painted just with the ethanol as a control, in case the alcohol itself, the ethanol itself, has antimicrobial properties. And then we, of course, put bees and combs in there, and then we marked the bees. And our goal was to study their immune system functioning. Okay, but mostly I wanted to just talk about the methods that we used to create a propolis envelope. The second method that we used later on, and this was done by another graduate student, Renata Borba, instead of painting the propolis on, we wanted the bees to create their own natural, quote, natural envelope. So what she did was take these propolis traps that are sold commercially here. I imagine they might be sold in Sweden, I don't know. But they have about 7mm gaps in the plastic, and they're about 7mm or even sometimes thicker in depth. So she cut them to fit inside of bee boxes and stapled them in. And the bees with these gaps stuff them full with propolis. So this photo is when she removed the traps, just to show the propolis envelope that the bees made with the traps. For our experiments, we always left the propolis traps there, and the bees would continue to fill them completely. This method kind of makes it difficult to manage the bees because the bee space is compromised a little bit, but for the experiments, that's what we did. And then what we did was collect bees of 7-day-old bees, all the same age. They were age marked, and we did real-time PCR to look at their immune system functioning. We wanted to know if propolis was affecting their immune system directly. And what you're seeing in all of these are names of antimicrobial peptides or other proteins produced or other enzymes produced by the bee's individual immune system. And every time you see this dark red, it shows that bees that have a propolis envelope have lower production. This is lower, lower, lower. Even if the graphs are upside down, it's lower than the controls. They have lower production of the immune system, which at first for us was very surprising. Why is it suppressing? Is the propolis suppressing the immune system? Then we realized that the bees within a colony with a propolis envelope, not only was their immune expression lower, but it was more constant over the season. It was less variable. And the interpretation is that bees are not having to invest as much energy in their immune response, probably because this propolis envelope is reducing the number and amount of, the amount and type of opportunistic and pathogenic microbes within the nest cavity. And let me show you, give you a human analogy that might help explain this. So maybe if there was mold in my house, I would be here sneezing because I have a slight allergy to mold. And if I would paint my house with propolis, all my walls inside my house here with propolis, that would kill the molds. The propolis would kill the molds. If we measured my immune system before my propolis paint, you would see my immune system elevated because my allergic response is producing an immune response to the mold. And after the propolis, after it killed the mold, my immune system would be quieter again. So we think that's what's happening with the bees, that the propolis envelope and the antimicrobial properties are killing off these other bacteria and molds and yeasts inside the nest cavity itself. So the bees are not having to invest energy in their own immune response. So we know that bees are not eating the propolis, but we wanted to know if the presence of that propolis envelope could possibly affect their microbiome, their microbiota. And we first started looking at within the gut microbiome. So here's the honeybee gut, their crop, ventriculus, intestines, and rectum. And each of these areas or these niches within the gut have certain microbes that have been very well described. What we found when we compared bees taken from colonies with a propolis envelope and compared their microbiome to bees from colonies without a propolis envelope is that the core gut microbes were more abundant and there were significantly fewer opportunistic and pathogenic microbes in bees with the propolis envelope, meaning that the propolis envelope, again, must be helping reduce all of these pathogenic or opportunistic microbes, allowing the core microbiome to flourish. Another student, Holly Waldalenberg, in collaboration with Keith Anderson, Dr. Anderson from the USDA Bee Lab in Tucson, Arizona, we looked at just the mouth parts, which is an interesting thing. So the microbiome is not just inside of us. We have microbiome all over us, on our outside of our body, in our skin, and inside our body, everywhere there's microbiome. And if you think about the bees' mouth parts, the mandibles and the proboscis, bees get their mouth parts into everything. So they clean the cells, and they feed each other. Am I okay now? Can you hear me?
Yes, we hear you.
Yeah, we can hear you.
Okay, good. Okay, sorry. All right. This Zoom is so bizarre, right? Anyway. Okay, so bees get their mouth parts into everything. They're feeding each other, they're cleaning cells, they're foraging, they're putting their proboscis into flowers, they're scraping these resins. So we were curious what the microbiome would be like on the proboscis or in the mouth parts of bees. So we collected bees and froze them and then clipped off their mouth parts and sequenced the DNA, sequenced actually just the bacteria that were on the mouth parts, again from colonies with a propolis envelope and without a propolis envelope. These species in green that you see, the OTU stands for Operational Taxonomic Unit, kind of equivalent to species of bacteria. You can see there's a lot more of these 3 bacteria in the bees with the propolis envelope compared to those without a propolis envelope. And these other in blue are these pathogenic and opportunistic bacteria. So again, just on the mouth parts, you can see these 3 species of bacteria that are comprise the core microbiome of the mouth parts and different from the gut. They were much more abundant than bees without a propolis envelope. So again, the propolis is helping their microbiome inside their guts and outside their bodies and on their mouth parts. Then we wanted to know if we challenge colonies with a pathogen, if we make them sick, and they have a propolis envelope, what's the effect? So in the first experiment, and these are fairly small experiments, this one is with 6 colonies in each treatment, we challenged them with chalk brood, which is caused by the fungus Ascosphaera apis. And again, these experiments done earlier were done with the propolis envelope that was painted on from the extract. And to provide the challenge, we took chalk brood mummies, shown here, and homogenized them and mixed them into pollen patties, pollen supplement, and fed them to the bees. And after about 2 weeks, the colonies get chalk brood. And then we quantified how much chalk brood they got. And colonies with a propolis envelope on average had 15 chalk brood mummies throughout the nest total, and without an envelope, 108. So it shows you the propolis really helped reduce the disease load. It didn't eliminate it. It's not a complete cure, but it helped reduce the disease load quite a bit compared to colonies without a propolis envelope. I'm sorry, I'll do it this way. And then Renata took it a step further, and we challenged them with Penibacillus larvae, the cause of American foul brood. And of course, after we did these experiments, we had to burn all the colonies so that we wouldn't contaminate. So we did it in a very isolated location. And again, fairly small experiment, 5 colonies each treatment. And this time the colonies had that the propolis traps inside. So the bees were making their own propolis. And we compared those bees to colonies without a propolis envelope, and we sprayed sucrose solution with a known amount of spores of Penibacillus larvae onto the combs. And what we found again, so these colonies in this red color had the propolis envelope and were challenged. And you can see they had significantly less American foul brood, and the severity of it was much less than colonies without a propolis envelope. Again, the propolis envelope did not cure the colony of the disease, it reduced the disease load. And a severity score of 3 is extreme, and 2 was, if you look at each frame, there's over 20 cells per frame with signs of American foul brood, and 1 is only 5, maximum of 5 cells per frame with American foul brood. Still bad, still infected, but not as bad as a 2. In addition, when we looked at the immune system of those bees that had been challenged, their immune response this time was in bees with a propolis envelope was higher than it was without, meaning that after challenge, the immune system was activated. And this helps us understand that the propolis is not suppressing the immune system. It's just keeping it quiet when they're not sick. And when they are sick, when they're symptomatic, they're able to activate their immune systems to help fight off the disease. Okay, so these data show us that the propolis envelope provides a therapeutic defense against chalk brood and American foul brood pathogens. In these studies, we found that it did not lower the levels of varroa mites or nosema or viruses. However, other researchers have observed effects of propolis on varroa and virus levels, particularly of deformed wing virus. So these 2 experiments from Nora Drescher and Garrido showed some slight effects. And this one was, the Garrido one was done in the laboratory. This one was done in colonies, natural colonies outside. So this for us is, makes us continue our research on the effects of propolis on viruses. And in the lab, we have a cell culture, the AME 711 cell culture. These are honeybee cells that we can grow in the laboratory. And we're currently, as we speak, in fact, next week, we'll finish the experiment, testing if propolis is active against acute bee paralysis virus in this cell line. Our previous experiments using this cell line showed it had no effect on deformed wing virus, but we're also retesting that. So stay tuned for those results. We know that propolis is active against human HIV virus. We have a publication from the graduate school, the medical school here from 2005, showing that propolis in human cell cultures in the laboratory is active against HIV. So we have some indication that the propolis may be active against honeybee viruses, which would be very exciting. Then we asked, okay, we've made these bees sick, and it was not fun to do, but it's part of the research. So while they're sick, do they collect more resin? Do they send out more resin for foragers? So are they self-medicating, or more correctly, are they social medicating? Because of course, the diseases that we're giving them are brood diseases. They're not adult bee diseases. So if they forage for more resin, it would be to help the brood and possibly the adult bees that are the carriers. So to do this, it's kind of tedious experiments, but we set up again some small colonies, and this is Renata Borba, she's originally from Brazil. We set up these small colonies with propolis envelopes or not, and before they were sick, we would sit in front of the colonies twice a day, close up the entrance, and we would count the number of resin and pollen foragers returning in a 15-minute time. We do that twice a day for 2 weeks. So here's a resin forager, here's a pollen forager, and you have to have pretty good eyes, so you need a lot of young people to help you do this. And then, excuse me, then after we have a count of what their 2-week foraging is, then we challenge the colony, either with chalk brood or American foul brood again. And then when they're sick, we repeat the counts. So we, again, twice a day for 2 weeks, we count the number of resin and pollen foragers returning. This photo shows you how tricky it is. This is a resin forager here. This is a pollen forager. Here's some more pollen. So again, you have to have really good eyes to see those. But what we found, and I'm just showing you 1 year of the data, but this we found every time, that when we challenged them with chalk brood, which is Ascosphaera apis, the fungal pathogen, they did increase their foraging for resin. This is after the number after minus before. They significantly increased compared to the controls that were not challenged. But when we challenged them with the bacterial pathogen, American foul brood, Penibacillus larvae, they increased resin foraging somewhat, but not significantly different than the controls. And we repeated this 3 or 4 times and got the same result every time, which was fascinating, that a fungal pathogen would lead them to collect more resin, but a bacterial pathogen not necessarily. And we don't know why. Then another student, Mike Wilson, we're working with some plant chemists. We sent him tree climbing. So we had him go collect tree resin from 14 species on our campus that are all around the foraging area of our bees. He collected that are known to produce resin. So he collected from five pine species, 3 spruce, 3 fir, 2 populus, and horse chestnut, what we call horse chestnut. And so he collected resin from those trees. And then we also, from the previous experiment, when we found resin foragers, we would collect those bees, chill them on ice, and remove the resin load from those bees. So we would put them in these sterile tubes here. And then we used reverse phase liquid chromatography and mass spectrometry to analyze the metabolomic profiles of these 2 resins, which give you these fingerprints that look like this. And you can match them up, and using principal component analysis and other statistical analyses, you can figure out if they're the same. So we can match the tree with the resin from the bee to figure out what the plant source is. And we found, so these are some of them, about Populus deltoides, Populus balsamifera, Populus hybrid. And these were actually the species that the bees collected most of their resin, but mostly it was from Populus deltoides, which is the eastern cottonwood tree. And if you have cottonwood trees in your area, you probably do, or some kind of Populus species, if you collect the leaf buds this summer and smell them, you'll smell the propolis probably from your area too, because most of the temperate, northern temperate regions, the bees are collecting resins from these particular plants. It was fascinating to me that we never saw bees in our area collect from pine or spruce. Maybe they do in your area, but not in ours, which was fascinating. So in sum, propolis is really good for the bees. It provides everyday benefits to their immune system. It helps promote the core microbiome species. It helps them reduce their disease load. Bees collect more resin, forage for more resin when they're infected with fungal but not bacterial brood pathogens, which is interesting. And bees in the Midwest United States, northern Midwest United States, collect mostly their resin from cottonwood leaf buds. So right now, in addition to the work on viruses, we're also interested in how we can help colonies and beekeepers, how we can stimulate bees to create a natural propolis envelope so we don't have to use these propolis traps. And we began by just scratching with a wire brush the interior of boxes. We also built some boxes with unfinished kind of rough lumber. And we found that, yes, they do put some propolis in, but it's not very much. So now we're working with a carpenter who's building boxes and creating deep grooves that are very rough inside the boxes. This is what they look like new, and this is what they look like when the bees have been in them for a little while, and they start putting the propolis inside these gaps and grooves. And we're testing right now how rough is enough. How much do they need to put in the colony in these boxes to support their immune systems and their health. So after leaving the bees in the box like this, these rough boxes for a while, we collect bees and we're measuring their immune systems to compare with when we gave them the traps and the extracts. We hope to have these data out this year sometime. So I want to finish there, and I wish I could be there to actually shake your hands, but I'll say thank you, and I imagine you have a lot of questions. So I'll stop sharing my screen so that you can see me and take questions. Thank you.
Okay, you can hear me?
Yes, I can.
Okay. Yeah, thanks a lot for that presentation. Yeah. We are a small amount of people here. The board actually from the association. The other ones are on the link. We have got some questions on the chat, and one main question is a little bit practical, I think, and practical. It's when we are coating plastic frames, we do it with wax. Shall we use propolis instead of wax, maybe?
No, I don't think so, because as you know, we really don't know how much propolis they're putting in the combs themselves, especially in the brood combs. Propolis can be toxic to bees. So we have some data inside the laboratory when we give bees in their very close proximity to propolis and to larvae, it can be toxic. So it's a medicine, and the dose makes the poison, of course. So too much can be not good for them.
Can be too much.
Right. Can be too much.
Okay. We also have a question about sharing the presentation you have done here. Is it okay to do that in Sweden, or?
Yes, I did send it to you, but I want to fix my typos.
Yeah, but we can live with that. But it's okay for you if you spread it.
Yes, that's fine.
That's good.
If you please, of course, credit us.
Yeah, sure. Any more questions?
Yes. Could you comment on wooden boxes and styrofoam boxes? Do we get less propolis in the plastic boxes? I believe you could be against these boxes.
Right. Well, we don't use plastic boxes, so I don't have any information about that, but I'm not totally against plastic boxes. I prefer a wooden natural box. I prefer to let the bees nest in a material that's natural to them. But I think if the inside of a plastic box was rough and had grooves, because like the propolis traps are plastic, we know that they would fill them with propolis.
Could an old tree benefit the propolis coating? A win-win. Feral bees have a home for longer time.
I'm sorry, I didn't quite catch it.
Could an old tree benefit the propolis coating?
Oh, yes. Yes. If you think about the inside of an old tree where the bees nest naturally, that is that's the kind of texture that they love to fill with propolis. So I suppose, I don't know if this is what you're asking, but I suppose you could take some inside of a tree bark and stick it in the colony and they would make propolis on that, yes. Excuse me.
Does the propolis contain at itself any sickness?
Oh, propolis can accumulate heavy metals. This was a study years ago by Bogdanov. I think he was from Switzerland. He did some analysis of propolis and found that it did have heavy metal contamination. We are looking at pesticides in the propolis too. Does it accumulate pesticide contamination? Remember that the bees are not consuming the propolis. They don't eat it. So if it is contaminated, they're not going to get it internally. Of course, by contact, they could become contaminated that way. But I think the pollen is more contaminated than the propolis, but we are looking at this.
In other words, should old propolis be removed?
No, you can keep the old propolis. It does lose antimicrobial activity over time, not completely. Renata Borba in her study collected propolis in October from colonies, and then from the same colonies collected propolis again in May, the following May after winter, and then did the test in the laboratory to see what its antimicrobial activity is. And she found that it lost some of its antimicrobial properties. It was still somewhat active, the older propolis, but then again, by May and June, the bees start foraging again and they pile more propolis on top. So I would say that if it's in your way, if it prevents you from managing your colony, you can get rid of the old propolis, but not the new propolis. I would encourage them, I would let them and encourage bees to bring in propolis.
Thanks a lot, then. Another close by question, I think. Do you have any idea about the wax situation in US at the moment? We have a lot of problems here in Sweden with or in Europe at all for wax fraud, especially with paraffin or stearin acid. And in some cases, a lot of dead broads because of that. Do you know about the situation there?
Wax fraud, in other words, selling wax that's not really beeswax?
Yeah.
No, I don't know about, no, that's a new one for me. I haven't heard about that.
Take care of what you have then. Yeah. Because it's a big problem in Sweden or the whole Europe at the moment.
That's horrible. I'm sorry.
Yeah.
I'll look into it. Thank you.
Mm-hmm. Okay, Marla. Thanks a lot for taking part of this Congress we have here, a little small digital one. We're gonna maybe make a bigger one next year with people around us instead of far away.
Yes, I hope everybody stays healthy, and I hope you all get vaccinated very soon.
Yeah, we're on the way here with some American, I think, vaccine at all, Moderna, for example, states. Okay, thanks a lot. I'll call you back in the week and we can talk further on. Thank you.